Another question that we come across is where we have to identify different potential differences like those given over here. As you can see, this one, we have X which is representing a point of minus 70 millivolt potential difference, and then Y which is plus 40, and Z which is minus 90. And it is said up here that table shows the potential difference across the neural membrane at 3 sequential time points X, Y, and Z. So sequential, the word sequential is a hint for us that okay, minus 70 is going to come first, then plus 40, and then minus 90. It comes in a sequential way. Now if you look at it, minus 70 is always responsible for being the resting potential of a neuron. Now how that is being achieved? They said describe the events. So the first event is resting potential. How are we going to describe it? Simple. First you'll talk about sodium-potassium pump being there, which is pumping 3 sodiums out for the sake of 2 potassium in. So overall more potassium diffuses out than sodium diffuses in. Now here you'll say why potassium is moving out, though we said that here potassium is moving in, but the potassium that is moving in is through sodium potassium pump, while the potassium which diffuses out is the potassium which goes out of the neuron through facilitated diffusion. And we have more channels there, so more potassium goes out than compared to sodium that is going to come in. That is how resting potential is maintained. Secondly, plus 40, or in some topics it is written plus 30. So plus 30 or plus 40 both represent the timing of depolarization. That is when a neuron is depolarized, and that is when voltage gated sodium ion channel opens up and sodium moves in order to make potential difference reaches to plus 30 or plus 40 millivolt. The third one is minus 90 millivolt. Now a lot of students do not understand this part, but this is about hyperpolarization. Remember, after depolarization what occurs is repolarization. That's when plus 40 is brought back to minus 70 millivolt. 90 millivolt represents the position of hyperpolarization. That is when your potential difference goes below minus 70, and it is because of potassium ion channels which remain open for a little longer, so potassium moves out of neuron and it brings the potential difference down to minus 90 millivolt.